Okay, we have a chromatography, and I'm going to show you how chromatography can be used to characterize proteins. Paper chromatography, I'll just write the word paper chromatography. Thin layer chromatography, what we call TLC. Yes, and then
HPL.
HPLC, what is the full form?
High performance liquid chromatography.
High performance liquid chromatography, HPLC.
Size exclusion chromatography.
Gas chromatography, size exclusion chromatography, SEC. Anything else? LCMS. LCMS is a liquid chromatography combined with the mass spectrometry. Anything other chromatography? Column chromatography, CC. There's a lot of branches. Anything else? Affinity chromatography, gel filtration chromatography,
Liquid chromatography.
HPLC again, high chromatography, high interaction chromatography. It's a lot of different chromatographic techniques. I just want to tell you that you already know many things about chromatography. Now, let's discuss about what we mean by chromatography and how the word chromatography came to come. Now, can somebody define what is chromatography? Separation based on color. That's a beautiful definition to have, because if you look into this word, chroma means color, graphy means graphs or writing, right? It's basically color graph is writing. Separation based on the color. Now I'm asking this question. Is everything colored in the world? Are proteins colored? Are amino acids colored? No, right? It's actually original definition, because back in the days, they tried to separate things based on the color. Especially, there was a guy, his name is M.S. Twist, right? He was, I think, he was Russian, Italian, or Russian Ukrainian scientist, if I'm not wrong. Around 125 years ago, he was there. And what did he do was that he took calcium carbonate, the chalk that we write on the board, right? And he made a column out of calcium carbonate, and he really wanted to separate what exactly present in these leaves, for example. You know, leaves is green in color, right? But it's not just green in color, right? For normal person, leaves are green. For scientists, leaves are green, but it's also green plus something, plus, right? And he really wanted to see what are the rest of the things present in this one. What did he do? He crushed these leaves and put it into a solvent. I think, if I'm not, ethyl acetate or something like that. He used a solvent, and then he loaded into this small column, the calcium carbonate column, like this one, right? Then he poured more and he loaded the sample. This is the extract of leaf. Then he added more and more solvent. Then he visually could see the separation of colored bands. Then he realized, okay, it's not just green. It's actually the extract contains too many other components. He could see orange color, he could see green color, he could see yellowish color, and lot of different colors he could see. Then we later realized that there is nothing but different components present in the leaf, right? We know that carotenes, carrot, orange in color, right? We know chlorophyll, green in color. We know xanthophylls, yellow in color, right? Basically, the leaf is mixture of different photosynthetic stuffs like chlorophylls, A, B, xanthophylls, and orange. And for naked eye, in the mixture, they look just as a green, but you can separate them in individual components, and then that becomes chromatography. Separation purely based on color. Then this was started in 1903, right? Now it is 2025. Thousands of experiments have been done. Numerous different things have been separated, and numerous detectors are attached to the chromatography, because our eye is the best detector in the world, right? We can do the eye test now. You can see the yellow color I can see, but our eye is not every time is right wants to see everything, right? Colorless compounds we can't see. Then we started attaching numerous detectors to see the unknown things that we cannot see in our naked eyes. And that becomes more complicated field. We call it as chromatography world. So I'm going to teach you a couple of chromatographic stuff, mainly thin layer chromatography. I don't teach you paper chromatography today at the interest of time. A little bit about column chromatography and mainly about HPLC. I think we can start with some fundamentals of chromatography, right? Whenever you take a chromatography, we have to remember about 3 different words. One is stationary phase, the second one is mobile phase, and third one is analyte. Stationary phase, as the word says, is static, doesn't move, right? Stationary phase. Now, in his previous experiment, calcium carbonate could be considered as a stationary phase. What is mobile phase? Exactly. The one that moves through the stationary phase is called mobile phase. Mobile phase can be liquid, can also be gas. In the gas chromatography, right? The mobile phase can be liquid as well as gas. If it is liquid, it becomes liquid chromatography. It becomes gas liquid chromatography. Okay. Is it clear? And the third one is called analyte. What is analyte? The solute particle that we want to separate, it's called analyte. I'm going to use these 3 words continuously in the future classes. When I say stationary phase, if something doesn't move. When I say mobile phase, if something moves. And the analyte is something that we want to separate. I don't define it again and again. Is it okay? Cool. So, as I said, the chromatography based on that one is divided into gas, liquid, thin layer and paper chromatography, and the column chromatography. They all come under the liquid chromatography category. And then HPLC, it's high performance liquid chromatography. What do we have here is that the stationary phase in thin layer chromatography often is silica. Okay, it's not necessarily always silica, but often it is silica. Please remember that silica is polar in nature. Silica is polar in nature, right? The mobile phase is organic solvent. It could be ethyl acetate, it could be hexane, and a lot of other organic solvents they use, right? This is usually relatively non-polar, right? One thing keep in mind in chromatography, polar likes polar, polar likes polar, non-polar likes non-polar, polar hates non-polar, non-polar hates, keep that in mind. Don't get confused with positive and negative. Positive loves negative, negative loves positive, polar loves polar, non-polar loves non-polar. This is the interaction you have to keep it in mind in chromatography. Okay. Now, what they do here is that they take a chamber, it's a glass jar basically, and they take the solvent that you want, mobile phase, right? And then take the thin layer paper, like this is a thin layer paper, and we just make a mark here like this, right? And we make a drop of your sample, 1 drop, right? 1 drop of sample is placed here, and then it is put inside the chamber like this. You should not touch the drop. Now what happens? The solvent moves against the gravity because of the capillary action. The solvent moves above the against the gravity. When it is moving, it will start solubilizing the analyte, right? When it is moving in this direction, it will start solubilizing the analyte. Now, this analyte, for example, is mixture of 3 different components, right? It contains yellow component, red component, and the blue component, right? Now, the solvent is moving in this direction. Initially, it will start dissolving this component, like, and slowly the solvent moves in this direction, right? Who will move faster? The solvent is moving faster because solvent is non-polar. It doesn't like the stationary phase. It just want to get rid of stationary phase, so it's moving faster. When it is moving faster, it will take its friends. Can you come with me? And these are the analytes, right? Now, the yellow one does not like stationary phase as much as possible, so it loves the mobile phase. So it moves along with the mobile phase. The red one, 50% it loves the stationary phase, 50% it loves the mobile phase. It moves slowly. The blue one, it really wants to stick to the stationary phase. It hates mobile phase. Mobile phase is pulling it. It says, I don't want to come. Based on this mobility, you get a separation. That's it. That's a thin layer chromatography. Purely based on the love for the stationary phase and love for the mobile phase. The interaction, the affinity. Clear? Whichever likes the one, it stays with that. If the analyte loves the mobile phase, it goes with the mobile phase. If the analyte loves the stationary phase, it tries to stick to the stationary phase. But there is a difference for this love, and that makes the separation. Is it clear? It's a fundamental of chromatography. The same knowledge is applied for paper chromatography, HPLC, many things. It's just about the affinity. Is it clear? Yes, welcome back to the afternoon session. And we began with the thin layer chromatography just to showcase you how exactly the chromatography works. We heard that polar likes polar, non-polar likes non-polar, and opposite does not exist. So polar hates non-polar, non-polar hates polar. And in a chromatographic system, there is always a stationary phase that is stationary, does not move, and there is a mobile phase that moves over the stationary phase carrying the analytes. Analytes are nothing but the ones we want to detect, separate, whatever, right? These are things we have realized in the last session. Now I have another TLC plate just to check your knowledge. So I have a TLC plates with the two types of amino acids I have. One is leucine, other one is glutamic acid, glutamate. Leucine is a non-polar molecule and glutamine is a polar molecule, right? I put this plate into a TLC chamber. Please note that the TLC plate is polar in nature. And then I started doing TLC of that sample. I got two components. I show it as red and green now. Now, tell me whether the red is leucine or glutamic acid. So, glutamic acid is polar. Stationary phase is polar. Polar likes polar. Where it wants to stay? With the stationary phase. It doesn't want to move together with the mobile phase. Leucine is non-polar. Mobile phase is non-polar. Leucine likes non-polar mobile phase. So it goes with that. It's a very important exercise to keep it in brain. It often confuses you. If you don't understand this basics of chromatography, your life will be tough in chromatography. We can't use thin layer chromatography for protein analysis. We can, but it's almost impossible. What could be the reason?
The proteins we deal in real life situation is a crude protein.
Yeah.
And so it will be tough to separate using TLC.
One of the things is that TLC is not so efficient in separating. It works with some of the organic, even organic chemistry people always use TLC to check the purity. But in protein science, especially in food science, we are talking about too many different things.
Protein is a complex system.
Protein is a complex, plus protein is heavier, right? So amino acid is a small, tiny one. It can move along with the software, along with the solvent. But the protein is a heavier molecule, and the capillary action perhaps is not sufficient enough to take that into the higher level, right? That is difficult to drag the proteins. You might get a small smear, then that's it. The third problem is that you need to see TLC always need a detecting agent. Either you can use a UV light or you need to spray something to see the color, right? And it's always challenging with protein sometime. Make sense? So TLC, don't try to use it for the protein science, but I used TLC just to give you an examples of how chromatography works. All right, so now the one we are going to use in food systems or in protein science is usually column chromatography. Usually column chromatography is basically the TLC knowledge can be translated to column chromatography. It works with exactly the same phenomenon, but in an opposite way. In TLC, we have a plate and we have a sample and we put a inside the solvent and things move in this direction, right? In column chromatography, you have a column and the stationary phase is filled in the column and the sample is loaded on the column, and then you load more and more solvent and the separation happens in this direction through the gravity. Sometimes you can also apply pressure and vacuum, but usually it's a gravity separation. Make sense? So in TLC, things go in the up direction. In column chromatography, it's like towards the gravity. That's the important difference. And this is how the column chromatography looks like if you want to separate some colored component from columns, just to give you an idea, right? And these columns can be made small, big, whatever size you need. And there is a lot of different types of stationary phases are available for separation and purification. Okay? All right. Now, I'm going to talk a little bit about how we are using this column chromatography to separate and purify proteins. Please note that chromatography is used not only for the identification and quantification process, it's also used to get high pure proteins. For example, if you want to get more than 90%, in addition to extraction, in addition to fractionation, in addition to alkali extraction, in addition to sometime dialysis, you have to use column chromatography. You can reach 95, 97, 98, 99% purity also sometime. You know that you can buy some milk proteins of 99% purity, right? Those can be usually purified. Of course, there are a lot of other technology. You can use column chromatography to get that 99% purity. That's the advantage here. Okay. Now this is just nice illustration I got, and you can see that imagine these are your proteins, and you have a lot of impurities here. These impurities in our plant-based world, it could be polyphenols, it could be anti-nutrients, it could be small peptide, it could be minerals, whatever, right? But we want only proteins separated, right? Imagine I just wanted to separate the green color protein here. How do we do that? And there's a lot of options. Let me take one by one. In general, column chromatography is nothing but you have a column and filled with some beads or some powders, and that is what we call stationary phase. Different numerous 100 types of different types of stationary phases are commercially available, and that is called stationary phase. And you put your sample in this range, in particular region, and then in the mobile phase, usually solvent. You can use different types of solvent here: polar, non-polar, aqueous, salt solution, alkali solution, different kinds of solutions you can use as a mobile phase, depending on what you want to separate. Okay. Now, I'll just show you one of the most important techniques what we use today to separate proteins and purify proteins. Is called size exclusion chromatography. Sometimes it's also referred to as gel filtration chromatography. Okay. Size exclusion chromatography, and I often say it as SEC or SCC, SEC. Okay. Size exclusion chromatography. As the word says, it is something, the separation principle is something related to size. Now, please listen to me carefully. It's an important chromatographic technique here to learn. In size exclusion chromatography, we have a column and filled with a special type of beads, right? It's filled with a special type of beads. I'll just try to illustrate here. This is the column, and the columns are filled with special types of beads, like this. Okay, and these beads are porous, means the beads are spherical, but they have holes inside. They're all porous. Or they are all porous like this one. Okay. Now, I want to separate rapeseed protein isolate, for example. Okay. Rapeseed has two kinds of protein. One is called cruciferin. Cruciferin. Its molecular weight is, let's say, 300 kilodalton. And we have another protein called nepin, and whose molecular weight is, let's say, 14 kilodalton. And it has a lot of polyphenols. Okay. Polyphenols having the molecular weight, let's say, 300 Dalton. 300 Dalton. These are all other kilodalton. Can you recognize there's a difference in the size here? The molecular weights are different, right? Now I want to separate them, these three separately. Now let's see how I can do this one. I have this matrix here, and that's a solution containing protein and polyphenol. This is the matrix, all right? I take it and I pour it on the column like this one. All right. Now I'll add some solvent and etc. thing. Now the sample is here, right? The sample contains cruciferin. I show it as a big bead here, big one. And the second, this one is nepin. And we have a tiny one is, let's say, polyphenol. Everything is here now. All right. Then I have a small, this one, I'm collecting the sample. Okay, this is me. Right. Now I want to collect the fractions. Now I load. Now tell me which fraction I'll get the first. Now listen to me carefully. Okay. We have the column filled with the beads. Beads are spherical, but each bead are porous in nature. They have small holes, porous in nature. Now you pour a solution containing different proteins of different size and a polyphenol with the smallest size in this case, for example, right? Now, the smallest polyphenol, what it will try to do is that it wants to come fast. I agree with that. Smallest and it wants to come fast. but it cannot come fast. Why? Because these beads are tightly packed. Instead of coming fast, it will pass through the pores. Okay, it will pass through the pores layer by layer by layer by layer by layer. Individual pores, right? It takes forever. The last one to come is polyphenol, the smallest one. And the nepin comes the second one because it wants to come in the porous, but there's also a gap between the beads, right? And the second one is coming the nepin. The last, the first one to come is the biggest one, is cruciferin. Why? Because cruciferin is the largest size. It wants to go inside the pore, but it can't go because the pore size is so small. Then you're still pouring the solvent on top of it. It tries to come in between the beads, right? It's the only case the larger one runs faster in size exclusion chromatography. Keep that in mind, right? It runs faster, so the cruciferin comes first. That is size exclusion chromatography. The bigger one comes first and the smallest one comes at the end. The biggest one comes first and the smallest comes at the end. Is it clear now? So our first fraction will be cruciferin, our second fraction will be nepin, and our third fraction will be polyphenol. And this is how you can actually separate multiple things using size exclusion chromatography. It can be made as a small column, an open column. It can also be made as a column for connecting to the HPLC. I'll talk about HPLC later. So you can make it in a different dimensions. And there is no interactions here to play, to be frank. No polar, those are very, very minor because we always try to use an inert bead. Okay, it's purely because of size. 99% I would say. Maybe there's small interaction for some other reason, but it's purely because of size. Is it clear? So in size exclusion chromatography, we can see it clearly that the bigger particle cannot pass through. It stays away and passes through the bead, between the bead, and it comes first. Okay, I have to quickly move on now. So we have another chromatography called affinity chromatography. It's also used quite often in molecular biology for the purification of certain specialized proteins. In food science, we can use it for purification of some allergens, for example. In this case, you also have a column, but this speciality of this column is that the beads containing some sort of ligands. Can you see? The beads contains some types of ligand, L, right? Now, if you add a protein, this is my protein, right? If the protein likes the ligand, it attaches with the stationary phase. Whatever that does not like the ligand, it just passes through. So it's a two-step process. You add the sample. If the protein likes the ligand on the stationary phase, it will just attach on the stationary phase, and the remaining things come out first. All right, but we want this protein. Can you see? To get this protein out, we add an elution buffer. It could be a salt solution. It could be another solution with high pH and so on. And then the interaction between the stationary phase and the protein will be broken, and the protein comes in the second step. Is it clear? It's a very simple way of doing things, but there's a lot of different types of ligands that are available. But the basic principle is the same. You have a stationary phase with the ligand, like this. Okay, stationary phase with the ligand. All right, and this is my protein, right? And I also have a lot of unwanted compounds, right? And I pour it on the ligand now, and the ligand likes the protein, so the ligand will attach to the protein. Other things will come down first. That is this step. Right? Now I actually want my protein also back at the end of the day. To remove the protein, I add another solvent, and the interaction between ligand and the protein is removed, and the ligand comes down in the second step. So this is called affinity purification. There's an affinity, is a love between ligand and the protein. Does that make sense? One of the beauty of column chromatography is that they can be made as big as this room also if you want. They can be upscaled to a crazy scale, provided you can control solvent and other things. Basically, you can see now this is one figure I've got it from the internet. The person is standing, you can see that the size of this column chromatograph, it could be of this size, it could be of this size, and so on. And sometimes the ligand, what we use there, the stationary phase, that you can reuse it. It's not that it's always use and throw. So you can reuse it several times, so you can make a high quality purification on a cost effective way. But I don't say it will come forever, but can be used for several cycles depending on the impurities present. All right, now I'll move on to the most important thing, is called high performance liquid chromatography, HPLC. In the column chromatography, we have columns with beads and we load here, and the separation is usually based on the gravity. So I collect the fractions, and who is collecting the fraction? Let's say it's a PhD student. And the person has to wait until every fraction comes. Do you agree? And since it's a gravity separation, sometimes it takes forever. Sometimes you will get drop by drop, 1 drop per second, sometimes maybe 1 drop per minute also. Then to collect that fraction, basically he has to spend half of his PhD maybe in some cases, right? So that is a bottleneck. That's a problem with the column chromatograph. I don't say it's a bad one. It's actually a slow process. But if I can make it faster, I need to flow things faster in this direction. Do you agree? From top to bottom in this case. So you apply pressure in this direction, or you have to apply vacuum in this direction. Do you agree? Either you apply vacuum or you apply pressure on the top. So basically applying the pressure on the top is always easier. So that becomes a high pressure column chromatography. And basically then the column need not be vertical always. You can put columns like this, anywhere, any position you can put, because either side is kept. Right? The stationary phase is inside and the solvent comes in this direction. And that is called column HPLC. It creates high pressure. Why? Because in this case, the particle size can be reduced to very small. When you reduce the particle size, you have more surface area. When you have more surface area, more interaction, more efficient separation. But the problem is when you pack the small particles so tightly, the pressure increases. Do you agree? The solvent does not have a space to pass through now because you densely packed the packing material. So the pressure increases. You can get a very high pressure depending on the type of packing, length of the column, and the viscosity of the solvent. The pressure can rise 300 bar, 400 bar, 1000 bar, 1500 bar, and so on. It's extremely high pressure I'm talking about. To handle that pressure, to create that pressure, we need an equipment that is called HPLC. It's also called high pressure simply because it handles so much pressure. It's also called high performance because now nothing is manual. Things can be made more automated. So that's called high performance. Is it clear? Right? The column chromatograph. Now I will, have you heard about HPLC and UPLC or something? It just, they are the same but different, right? Same mechanism of separation and everything. HPLC usually operate up to 400 bar. I might be wrong here because people have different claims for that one. UPLC can handle up to today's 1800 bar-ish, something like that. That's the only difference, right? You can even make a very fine particle based column with UPLC. Efficiency is very high. You can reduce sample separation time with UPLC compared to HPLC. Okay. But if you have more questions, you can ask me later. I'll not go into detail all these things. Just remember, they are the same but different pressure operation, maximum pressure operation. Okay. Now I'll briefly show you the different parts on the HPLC. Quickly show you. So HPLC has several parts, and the first one is the solvent reservoir, which is on the top of the instrument always. And basically, in the normal column chromatography, you have to pour solvents, right, manually. But here it's basically mostly automated. Whatever solvent you need, put it in the bottle. There's a pipe connection. It will do things most automatically, right? And then you have a most important part of the HPLC is called pump, because that is something handles high pressure and sends the flow in the right flow. If you want 200 microliter per minute, it will send exactly 200 microliter per second. And that's the job of the pump. It takes the solvents from the bottle upstairs, right? Okay. And then we have a sample compartment. In the column chromatography, you remember we have a sample, is one sample at a time. Do it manually again and again. But even here in this auto sampler can handle, like, let's say, depending on the type of the sampler, you can have 100 samples, 200 samples, 300 samples in an autosampler, and it's also temperature controlled. You put it in the vials and go home, everything will be done automatically. It injects automatically, it analyzes automatically, everything is automated. You just have to place the sample in the right place. You don't do a manual injection, you don't do a manual loading, so it's having an automated injector and auto sampler with the temperature control. Okay. And then the second important thing is here is the column, and the column comes with the different stationary phase type, which I'll take it in the next couple of slides. And what actually happens is that you inject the sample here, right? And the sample goes through the column like this one. Separation takes place, and then it comes to the detector. So HPLC can be connected to different types of detector. Remember, it could be UV detector, it could be fluorescence detector, it could be refractive index detector, it could be diode array detector. Is actually similar to UV. Fluorescence detector I covered. It can be detected to the most famous MS, mass spectrometer, right? So LC is the plot where the separation takes place. Separation takes place here, and once the analytes separate, they come one by one, and that will be detected in the detector. Am I clear? And these are different types of detector depending on what you want to measure. If your analyte absorbs in the UV region, you have take the UV detector. If your analyte is fluorescent, take fluorescence detector. If your analyte is having a refractive property, you take refractive index detector, for example, sugars, organic acids, and so on. So depending on what you need, different techniques are available for the detection. Now I'll talk a little bit more about the HPLC column. Okay, it's a very important subject. HPLC column looks like this. Have you seen it before? It comes in a different length, dimensions, and so on. An HPLC column look like this. It's usually made up of stainless steel. Yeah, one of the reason is corrosion resistance. Second reason inert. The most important reason, they withstand pressure. I don't say plastic column does not exist. They do exist, but usually stainless steel. It's simply because we're talking about 300 bar, 400 bar. It's actually high pressure. If it's a weak plastic, it will just burst off. So that's the reason why you use a heavy metal, steel one as the column. And the stationary phase is actually inside this one. You can see the white color silica if you open these two nuts, but don't try that one at the lab. But because each column can cost you approximately 80,000 rupees to 1.1 lakh, 2 lakhs, something like that. Your supervisor will be super unhappy if you try to open it. You're welcome to do it, but don't tell that I told you to do that. All right. So how the separation takes place in HPLC column. So the mobile phase is moving in this direction. Okay, mobile phase is coming from the pump. Okay, from the solvent reservoir, mobile phase comes to the pump, and the mobile phase is going in this direction. Okay, and meanwhile you have injected the sample here, right? This is the sample initially in the beginning of the column, right? The sample contains a lot of analytes. Let's say it has analyte A, analyte B, and analyte C, right? And the solvent is moving in this direction, exactly like thin layer chromatography, right? Now separation takes place exactly on the same principle. If the analyte likes the stationary phase, it wants to stay with the stationary phase for a long duration. If the analyte likes the mobile phase, it wants to go with the mobile phase. So that is why you get the separation. So different analytes as a band. Can you see? Blue likes the stationary phase. Now, at the end of the day, I get a detector. The detector gives you the chromatogram, which looks like this. Have you seen the chromatogram? Okay, this chromatogram is belonging to these three analytes now. These three peaks in the chromatogram belongs to these three analytes. Now tell me, among these three peaks, which one is blue? Do you agree the last one is blue? Yes, because the blue is retention time is longer. The retention time, and this could be the abundance or absorbance, whatever, right? So retention time is longer. It wants to stay in the stationary phase. It comes at the end of the day. So this is blue, and this is red, and this is the first one is yellow. This is the basic principle of HPLC. And you can buy different types of stationary phase to separate different molecules. the stationary phase varies. And when the stationary phase varies, the mobile phase varies. Okay. Now let's move on to the same thing. Polar lights, polar, non-polar lights, non-polar, positive lights, negative, negative lights, positive. Remember that one. Okay. Based on that principle, we have different types of HPLC, normal phase, reverse phase, and depending on the size, you have a size exclusion chromatography, ion exchange chromatography, hydrophilic interaction liquid chromatography, helic or helic. There are different types. I'll try to cover, I have already covered size exclusion for you. I'll try to cover these two. In the normal phase chromatography, the column, this is the column. Okay, this is the column. The column is filled with a powder, a very fine powder, and that powder is made up of silica. Okay, very cheap actually, but it's high quality silica. It becomes expensive, so it is filled with silica. And if you, this is the structure of that particular column powder, right? You can see that one. How does it look like? I would like to thank Waters here, because all the slides I borrowed from Waters, just to let you know. They had a nice, fantastic presentation for me ready, and I just used them. So just to let you know. So it has a hydroxyl group. Can you see? Okay. Hydroxyl groups are polar in organic chemistry you studied, right? So the stationary phase is polar here. Remember? Stationary phase is polar. This is how exactly the silica powder looks like. It's having a very fine particle with a high surface area. Okay. How the separation takes place in the normal phase chromatography. I have a sample that contains 3 different analytes, right? And the mobile phase here we have to use non-polar. Can you see? The mobile phase we have to use non-polar. We are not using polar mobile phase. Stationary phase is what? Polar. The mobile phase is non-polar. Now I have a question. Why can't we use a polar mobile phase? The biggest problem here is that if the mobile phase is polar, stationary phase is also polar, polar likes polar, stationary phase also likes mobile phase now. So now mobile phase wants to stay in the stationary phase, and you're continuously pumping in this direction, but it doesn't want to go out. What do you create? The pressure will increase like anything. So that is not a good separation to do. Am I clear? That's one of, you need to have a kind of an opposite effect to elude them. Exactly what she mentioned. Somebody should carry somebody. If everybody stays it, there's no separation. Is it clear? I have 3 peaks now. Which one is blue? The first one is blue, right? This is blue. Now I ask you a question, which one is polar? Blue is polar or yellow is polar analyte? Yellow is polar analyte and the blue is? Wow, I'm so happy to get this answer. You understood chromatography. Thank you so much. So that's the right answer. Why we don't use a normal phase chromatography for polar analytes, let's say sugar and polar free amino acids. What is the problem with the non-polar solvents? Exactly. The non-polar solvents are very toxic. We need to use hexane, for example, in this case. And they're toxic. They're not environmental friendly. They're highly volatile. People know that if you breathe it, it's a problem. And plus, non-polar solvents melt so many things, right? It melts plastic. It dissolves everything. So it's very difficult to handle this non-polar solvent in an analytical laboratory. So that's the reason why we don't use non-polar a lot nowadays. Only if in exceptional cases we use. Make sense? To avoid all these things, we have a reversed phase chromatography. As the word says, it's the reverse of normal phase. Okay? Reverse phase is just the reverse of normal phase. In this case, the stationary phase contains silica, but that hydroxyl group is blocked by a non-polar thing. Can you see? The hydroxyl groups are blocked by a non-polar thing. Have you heard about C18 column? You often hear the C18 reverse phase column, C18 reverse phase column. It's nothing but the carbon is, total number of carbon is 18. Right? You can see 1, 2, 3, 4, 5, 6, 7, 9, 10, 12, 13, 14, 16, 17, 18. Right? So this is C18. That gives you the non-polar nature to the column. Please remember, in C18 column or in the reverse phase column, the stationary phase is non-polar. Now let's move back again here. In case of reverse phase chromatography, stationary phase is non-polar, mobile phase is polar, right? Now you tell me which one is the most polar analyte here. Yellow will come first. Fantastic. It's just opposite of normal phase chromatography. That's it. There's a lot of variabilities we can use to maintain. I explained it in the most simplest way as possible, but it's a very complex system, HPLC. You need to play with too many things. You need to play with the column size, type of the mobile phase, the gradient program you're using, temperature of the column, what is the mobile phase you're using, and what in which solvent your analyte should be dissolved. There's a lot of things you can change in HPLC chromatography, in HPLC, to get the best separation. Please note that for a chromatographer, we always love to get peak like this, a Gaussian, well-separated peaks. This is the dream of a chromatographer. In reality, we get like this, and too many problems we have when you start chromatography first. But when you optimize all these things, you can bring this to this one. Make sense? Peaks can be broad and peak can be tailing, peak can be fronting, the peaks could be split. A lot of problems you will see. By optimization, by changing all these things, you will get a nice chromatogram. Okay, is it clear? This is something, the job of the chromatographer in your laboratory. And usually, if you go to any chemical laboratory, food analytical chemistry laboratory, there will be a dedicated person handling the HPLC. You have seen that one. They say, don't touch the machine. Oh, I don't want to give it to you. It's simply because it's not that they don't like you to train you. It has a lot of parameters. If you do a small mistake, things can go really wrong. For example, for HPLC column, they're tightly packed. If you forget to filter the samples properly, what it will happen? It will block the column. As I said, the column can cost you all the way to 1 lakh Indian rupees. And I don't want to throw that column, right? It's so expensive. Sometimes we put the guard column to prevent all these things. Still, the guard column is also expensive. So it's a lot of money to run an HPLC. HPLC is also a very, very nice equipment, but it's also very expensive to run. Just remember, okay? Compared to TLC. TLC could be 1 rupee, HPLC could be 1,000 rupees, the price I'm telling about. All right. Now, I want to tell you one important thing. HPLC can be used for identification as well as also for quantification. Why? Because the area under the curve is directly proportional to concentration. The area is directly proportional to concentration. By injecting the known standards at different concentration, you can plot a calibration curve like you do it in the UV that you've done in a laboratory, right? You can make a calibration absorbance versus concentration, area versus concentration, perpendicular, you get the concentration of the unknown compound. So, HPLC is often used for the quantification. You can quantify how much protein is present. You can quantify how much amino acid is present using right type of chromatography. Does that make sense? So, the peak area varies depending on the concentration of the analyte. Okay. Please note that I'm only showing the basics of chromatography considering the students here, but just to let you know. But if you need an advanced knowledge, please come and talk to me later, okay? Now, just to show a very nice example how the chromatogram of a protein look like. It's from the milk, not from the plant. In milk, you have caseins and beta-lactoglobulin and all these things. This is purely based on reverse phase chromatography. Now, based on this, can you tell me among these proteins, which one is polar? Kappa casein is polar, right? It's polar, it comes first. It doesn't like the stationary phase. It has a shorter retention time, right? And this is the most non-polar analyte. So that's how we can separate based on the difference in their polarity, because every molecule will have a difference in polarity. And that is the advantage. That is the thing we leverage here to get the separation. Now let me come to the plant protein world here. You can see how dirty the chromatogram look like. So it's not a nice chromatogram according to me, but still you can separate certain things. And basically the B and C are conglycinin and glycinin peaks in foil isolates basically. So you can actually separate them. You can actually identify them and also quantify them. And I can show you more dirtier chromatograms here for plant-based one. So it looks really dirty, right? But people try to get more information from this. Please note it's not the fantastic chromatograph in the world. If you do a really nice chromatogram, it will look like this one. Very sharp, nice Gaussian peaks. But proteins will never behave like this. This is amino acid, free amino acid. The proteins are crazy. They interact indifferently. look like so crazy. It's very difficult to integrate this peak and find out the concentration. Even software cannot do it properly. Am I clear? But still we can use them for identification and quantification. What I want to tell here is that you can use reverse phase chromatography to separate proteins by HPLC, but don't think it's an easy go. It's very complicated because separation is always compromised. This is another nice example of size exclusion chromatography they are using to separate what was that is in pea protein concentrate, and they have legumin, convicillin, and vicillin, and so on. These things. It's a size exclusion chromatography. Now tell me which one is having the largest size. So in size exclusion chromatography, the bigger one runs first, faster, so it comes out. So this is having the largest molecular weight, and this will be having the smallest molecular weight. And this is actually very complementary to your STS page. Remember my previous class STS page? The STS page, you get the last band here, and that will come here. Okay, the big one will come here. So we often use size exclusion chromatography complementary to STS page. You can also run molecular markers here as a standard to find out the apparent molecular weight. In this case also. So we run these two in parallel to see the apparent molecular weight of unknown proteins or known proteins in plant-based or alternative protein. I really recommend this technique in case you want to use for the future. So here is the separation for size exclusion chromatography using separation of rapeseed proteins using size exclusion chromatography. Rapeseed has cruciferin and it has nepin. Cruciferin has 300 kilodalton. Nepin is approximately 14 kilodalton. Now tell me which one is cruciferin and which one is nepin here. First one is cruciferin, right? Bigger one comes first. Nepin, right? We can also use HPLC to separate amino acids. After hydrolyzing the proteins, you can separate individually, you can identify and quantify the amount of amino acids. You remember in my second or first lessons, I talked about a lot of essential amino acid present, a lot of non-essential amino acids present. All of them were measured by either HPLC or GC. Exactly the same technique, right? For example, you have a glycine, we have a methionine somewhere here, tyrosine, and so on. So all these are identified and quantified based on chromatography. The area under the peak is directly proportional to concentration. Okay. In the previous picture, we had an HPLC with a column, right? And we are pumping the solvent like this one. We are injecting the sample like this, and the separation takes place, and things come in this direction. And there is something called a detector here, right? And the detector will detect the ions, and I said you can use UV, you can use fluorescence, and so on. And then you get a chromatogram. This is what I explained you past 1 hour, chromatography, right? Now, we can use another type of detector here that is called MS, right? This is liquid chromatography. Can you see? This is liquid chromatography, LC. And then you have an MS detector, and that becomes LCMS. Have you heard about this word? So simple. Please note that in the LCMS, the left-hand side is the same as HPLC or UHPLC or UPLC. Now, only the right-hand side, what I mean here, the detector is different. Is it clear? Now, I'm going to teach you the MS little bit. At the interest of time, I can't go too deep into the MS. Please note that MS is again very complicated thing in the world, I would say. It's a fantastic equipment, but it's a lot to learn. How it operates, what is mean by MS spectra, and how do we understand an MS spectra, and how do we get the information out of MS spectra. It's a little bit complicated. I can't teach you it in half an hour. It is basically a class of a GAN program itself, according to me. It's a lot. But I'll try to cover, considering the class background here, I don't go deeper into the subject. I'll try to give you the basic overview of an MS system and how we can use it for the protein characterization. Is that okay with me, with you? Okay? Yes or no? Yes. Cool. Now, we have been seeing that in the HPLC, separation takes place. based on the band, and then that reaches to the MS, and then you will get something called MS spectra. Okay, and that is the end product, the MS spectra. In between, there is a lot of things happens, right? You have an analyte is coming out of the column. Okay, then the analyte gets you the MS spectra. This is called MS spectra. But to get the MS spectra, there's a lot of things happening in between. I'm going to show you exactly what exactly happens here in between to get the end of the day MS spectra. In brief, in short, okay? Now, I need to answer this particular question before going to that particular topic. When and why do we use an LCMS? Can you tell me? Have you ever used LCMS? Yes? Or you must have at least supplied the sample to somebody to run the LCMS, right? That's often we do often. Why did you supply the sample and why did you run the MS? Okay, you get the separation based on the mass, but please note that that separation and the MS separation was different. The separation of the analyte happens in the column. Remember, the separation of the analyte happens in the column, but mass also a separator. It separates the analytes based on their ions. That I'll come back to you later. You're right. But if you're talking about this separation, it's different. Okay. Any other?
It's a derivative of that sample.
It gives a
Derivative of the same sample.
what that
Means carotenoids has lot of derivatives.
Okay, you get more
We
information.
can identify how many derivatives is formed during the reaction.
How do we identify? What extra information does mass spectrometer gives? It gives different peaks. Mass, right? Mass spectrometer gives mass. That is the most important thing we need for the structural elucidation. Do you agree? If I know the mass, exact mass of a compound, 50% of my problem is solved. I'm telling you. If I have an unknown compound, if somebody tells the exact mass of that one, 50% of my problem is solved. Then I'll go to an NMR person and tell you, okay, can you get an NMR spectra? Then I'll go to an XRD person and tell you, can you get me XRD? Then I know the structure of the molecule. Am I clear? So the mass is a fundamental thing we need to get the structure. And structure talks about composition. And the mass spectrometer is a fantastic thing in the world that talks about the structure of the molecule based on the mass. It also gives you the fragmentation pattern. And those fragmentation pattern, for example, this is the fragmentation pattern. This is the mass of the ion. It has a parent for fragment ions here. And this is unique for each molecule, right? In the mass spectrometer, let's say one example. Let's say you have a compound that is looking like this one. Okay? In the mass spectrometer, it will be fragmented in different possibilities, right? And each fragment will give you one kind of ions here. And it is very unique to that particular molecule. Now I can get you the more information about the molecule, its structure. I get the mass, I also get the fragmentation pattern, and that gets me the structure of the molecule. So structure elucidation becomes super useful with the mass spectrometry. All right, so another important advantage is that you can analyze few more topics based on, we can analyze a lot of compounds in a very complicated matrix using LCMS. But LCMS is super expensive, super expensive compared to HPLC connected to UV detector. I'm talking about 10-fold, I'm talking about even 100-fold expensive, right? In Indian rupees, we can talk about crores to get a mass spectrometer, a good one, right? It's actually an expensive piece of equipment, but it gives you the fantastic information. All right, these are the basic instrumentation. Since I don't have much time, I'll try to take it as a image here only. Is it okay? So I'll show you how the mass spectrometer works. You have a column here, and you injected the sample here, right? The analytes are coming out of the column. The one analyte came out, imagine the analyte A came out of the column, right? In the mass spectrometer, we have a chamber called ion source. This is the first important part of a mass spectrometer. And then we have another chamber called mass analyzer. Mass analyzer. And then we have a last one, D, that is detector. These are the three main part of an MS equipment, right? The analyte comes out of the HPLC column. The analyte just comes out of the HPLC column. It enters the ion source. Okay. Please note that the analyte that comes out of the column may not be charged. It could be neutral, right? Mass spectrometer can separate ions when the mass spectrometer can separate an analyte when they are ionized. Note that point. Mass spectrometer can separate analytes when they are ionized, right? So we need to first ionize them, right? That is the job of the ion source. So the analyte A, it's coming and entering the ion source. It will get a charge. The charge could be positive and charge could be negative, depending on what kind of voltage you're applying. It could be positive or it could be negative. Once it is in the charged, then it enters the mass analyzer. Mass analyzer is the one, basically it's a filtering system, right? It filters ions based on their mass by charge ratio, M by Z. Okay, it filters the ions based on their mass by charge ratio. I'll explain that in later in the next slide. So that's the job of the mass analyzer. Basically, when the ionization takes place, it's only you don't get a lot of different ions. You get A plus, and the daughter of A plus may be A1 plus, A2 plus, A3 plus. So many things will be formed in the ion source. But I don't want everything. I just want A plus. You tell the instrument. Then the mass analyzer will filter only A plus and sends everything out to the waste. That's the job of the mass analyzer. We don't need everything. We need what we want. You can tell to the computer, you tell to the machine, I just want A plus, rest everything should go to waste. Then it will adjust its magnetic and electric field so that it will select the ions based on their mass and charge ratio. And then it will come to the detector, and then you get the so-called mass spectra. Make sense? This is mass spectrometer in 2 minutes. It's so much. Now I'll talk a little bit about how the ionization happens. Please, I'll repeat once again, just keep this in mind. Analytes come from the column, right? Before you want to get the mass spectra, it has to be ionized. Ionization will be done by the ion source, okay? The mass filter or the mass analyzer will filter the ions based on their mass by charge ratio, and then the detector will detect those ions. That's a lot of detector, forget about that, it's not important. And then you get the spectra. Is it clear? The detectors are diodes and photo, there are many things, okay. Now, this is what I was trying to explain. It's a lot of different types of ion sources we have, okay. The first part, have you heard about different ion sources? Electron impact ionization, chemical ionization, electrospray ionization, atmospheric pressure ionization, atmospheric pressure chemical ionization, atmospheric pressure photoionization, MALDI. Okay, MALDI-TOF. There's a lot of different ways you can ionize your proteins or analytes. This part. Okay, this is a very typical unit. A number of different types of units are available commercially. Okay, depending on what you want to ionize, you can buy the one you want. The most commonly used is EI, electron impact ionization, especially in GC, and ESI, electrospray ionization, especially in the LC, and MALDI, and also we use in the protein science actually. Okay, I'll try to cover only these two today, EI and ESI. These are the most common used. And the mass analyzers are many: quadrupole, ion trap, TOF, orbit trap. You must have heard about LC TOF, LC Q TOF, LC orbit trap, LC triple quadrupole. There's a lot of mass analyzers are available, and they filter the ions differently. How you want to filter in this particular part, then you get a different piece of equipment. Am I clear? So depending on the source and depending on the mass analyzer, the price of the instrument also varies. Okay, mass analyzer is a very important thing. The source is a very important thing that gives you the high quality information. It's something, if you want high quality data, you need to buy the best mass spectrometer in the world, and that brings you the money. And everything has their own advantages and disadvantages. Is it okay? Now, I'll talk a little bit about ionization, especially electron impact ionization. Okay? Now, the analyte is coming here from the column, right? And this is an analyte, right? And this is entering the ion source now. It came inside the ion source. We apply a high voltage electron at high energy, right? And this electron hits the analyte, right? After hitting the analyte, it will knock off the electron from the analyte. When you knock off the electron from the analyte, you get analyte, radical, anion. Sorry, cation. Okay. Now, can you see you got a charge on the analyte now? Originally, it was neutral. Now, you got a charge. Okay. But the energy that electron hits is so fast that the molecule can be fragmented. Okay. So, you not only A plus you will get, you will also get the fragments of A plus. I'll write it as A1 plus, A2 plus, A3 plus, so on. Its fragments are basically, let's say, this is an analyte. It can be fragmented everywhere, okay, because of the impact of the electron. So basically, you get a different types of analytes, right? And these analytes are now pushed in the vacuum in a chamber like this one, okay? These analytes come to the vacuum chamber like, and here we apply the magnetic field. We apply the magnetic field. Are we running out of time? I need some 5-10 minutes at least. Okay. We apply a magnetic field, right? We apply the magnetic field. Now, you know that ions can be deflected based on the magnetic field. Ions will move in a different direction. They go crazy when you apply a magnetic field, all right? Now, we have ions coming, which is A plus and A1 plus and let's say A2 plus. Now, A1 plus is a bigger size, A2 is second big, and then your smallest one is A3, imagine, like this, right? And they deflect under the magnetic field in a different ratios because they have a different mass by charge ratio, okay? Usually, the smallest one deflects much easier than the biggest one, right? So, basically, the smallest one is A2, it will deflect more compared to A plus, the bigger one, right? Now, what we have is actually the spatial separation of the ions based on their mass and charge ratio. Okay, in the small, this all happening in fraction of second, fraction of second. Basically, it's happening so fast, right? So they separate in the mass analyzer in the here, and then they will be detected as an individual ions because they are separated here based on the mass by charge ratio. Either clear? And this is the basic principle of the mass spectrometry, the ionization and separation. So you have a high energy electron, it hits the analyte, then the analyte can get a charge. But it not only get the charge, sometimes it will also break the analyte into multiple pieces. Then you get a bigger fragment, smaller fragment, and so on. And all the fragments will enter the mass analyzer here. And under the vacuum, under the high magnetic field strength, they will separate spatially based on their mass by charge ratio. And at the end of the day, you can detect them as a MS spectra. Is it clear? Are you getting my point? Okay. Yeah. Now, just to show one example, you have phenylalanine. This is an amino acid. It can be fragmented into different pieces. Can you see? And this can move in a different velocity if you have a charge. And this can move in a different velocity. And this can move in a different velocity in the mass analyzer. And that can be used as a separation principle. You get a mass spectra, something like this for each fragment. Okay, this is mass. And then I'll briefly go through the electrospray ionization. This is called ESI, often used in the LCMS. It's a very similar to the previous one, EI. EI is the electron impact ionization. Now I'm talking about the electrospray ionization. It's often used in the LCMS, liquid chromatography, not in the gas chromatography. So what do we do here is that the analyte is coming again here from the column. This is my analyte, okay? And the solvent will have this solvent charge and so on. Basically, we spray, I hope that I have a figure that one, yeah. We spray the analyte in a small nozzle. You know the scent bottle? You spray, becomes an aerosol. Exactly that thing we do here. Okay, with some gas, we spray the analyte, it becomes an aerosol, right? And the temperature will be here in maybe 200 degrees Celsius, maybe 300 degrees Celsius. Okay, and then it will get kind of a small droplets of the ions like this, right? At certain point, the droplets will be broken, then you will get the individual ions with charge, right? Individual ions with charge, and that will go to the mass analyzer. Very simple procedure, but it has a lot of complicated machinery to do this one. So basically, you have an ion. In the liquid chromatography, you have a liquid, okay? The liquid is mixed with gas, and you actually spray them, and then there will be formation of the bubbles with charge. I don't go in detail about all these things, don't worry. Just remember that you get the ions with the charge. In ESI source, you get a positive charge by adding a proton to the molecule. This proton can come from the solvent. You can also you can remove the proton from the molecule, then it's called negative mode ionization. Have you heard about ESI positive mode ionization, ESI negative mode ionization? Now, that is something related to the structure of the molecule. If you have an amino group, it can easily protonated, so we run it in the positive mode, M plus H. Now you get the charge. Basically, it's the molecule plus a mass of the proton. The mass of the proton is 1. Okay. So imagine the molecule has some, in this case, the molecule has 721 as a molecular weight, and it added 1 proton, it becomes, sorry, 722, and you get 723. Okay. And in the negative mode of ionization, you remove a proton from the side chain, and it gets the charge. And the charged ions will again move in the quadrupole and so on, and they will get separated. Okay. I just have to skip this one. Now I'll just, there's a lot of different types of mass analyzer. Unfortunately, I can't cover it today at the interest of time, but I'll just give you how we are using this mass spectrometry in protein analysis. Okay. So we do the ionization of the proteins, right? And they are charged now. You can see different ions, and they are separated in the mass spectrometer, and you get individual chromatogram like this. And each chromatogram, it may be the peaks is associated with the protein, different type of protein, protein 1, protein 2, and protein 3, right? And under each peak, you have a mass spectra. And that will help us to identify the unknown proteins, because the mass of a protein is very unique, right? That will help us to identify. Not only identification, we can also use it for the quantification. Why? Because the area under the curve of each mass fragment is directly proportional to concentration. So this is called intact protein analysis. Intact protein analysis. We also call it as top-down proteomics. So basically, the analytes are ionized and supplied to the mass spectrometer, and you get a peak and a chromatogram and a spectra. You can use it for the quantification or also identification. Then we have a peptidomics, or what we call bottom-up approach. In this case, what we do, we don't inject protein directly to the mass spectrometer. Before injecting the protein, we hydrolyze the protein into individual peptides. Then the size of the protein is shrunken, right? Then you get a peptide fingerprint, and that will help us to understand which amino acid is hitting one another, because each peptide will have its own fragmentation pattern, and we get another information here. By combining these two information, we can talk about the structure of the protein, the molecular weight of the protein, the sequence of the protein. And these are the two approaches we are using to do protein characterization in food science. I only took it only half an hour to explain mass spectrometry. As I said, It's a complicated field, and I really need another 6 hours to explain this in detail for you. But that is not necessary considering the background of the classroom. Okay, we are coming toward the end of the lecture. So I just wanted to mention about mass spectrometer. Just remember that the mass spectrometer is nothing but the instrument that measures the mass by charge ratio, and that is an important tool to identify and as well as quantify proteins, amino acids, whatnot. It can be used for almost everything. As long as the molecule ionizes, we can use it in the mass spectrometer unless they're extremely big. Okay. Principle is simple. There is an ion source where the analytes will be ionized. Only ions can travel in the vacuum under magnetic and electric field. Okay. And there is a mass analyzer next to it, and it will filter the ions based on the mass by charge, and then we will get a detector that will detect it as a mass spectra. That's the basic principle of mass spectrometry. Okay, that's it for today. Maybe today, to conclude, we have studied too many things. I can say that today we covered about spectroscopy. Can you mention which one we covered to begin with? We started with primary structure of a protein, Edmonds degradation, secondary structure of protein with FTIR, tertiary structure with fluorescence, and also with XRD, and then STS page for the separation and molecular weight determination. Then we had learned a lot about chromatography, the basic principle of chromatography, polar, lights, polar, and so on. TLC, column chromatography, size exclusion chromatography, reverse phase chromatography, normal phase chromatography, affinity chromatography, and then we came to mass spectrometry. When you get a slides, just go through the slides once again. If there is a question, get back to me. With that, thank you so much.